Okay, good morning everyone. And in this work, this is an adventure into multiple brain subnetwork excitatory inhibitory balance. So why multiple brain subnetwork? Because a comprehensive assessment of multiple brain subnetwork in single subject has been rarely performed. So we decided in this work to focus our attention on this, the network that you can see here. So the visual network, the somatomotor, the attention, the limbic, the frontoparietal, and the default mode network, and to try to understand the excitatory inhibitory balance in each of these networks for each of our subjects. So in order to do so, we took advantage of the sequent workflow, so starting from diffusion and resting state fMRI of each subject, we follow the subsequent workflow, and we perform the virtual brain simulation in multiple networks, taking advantage of a model, which is the so-called one-one model, which enables us for each single network to explore the excitatory inhibitory balance in a personalized way. Why? Because thanks to the one-one model, we can focus our attention on 4 different parameters, which enable us to understand the excitatory inhibitory balance. So having a flash look at the results, we have 3 main results for this research. First, the virtual brain parameters presented differences between all the function networks that we analyze in our work. So this suggests the presence of a specific network-dependent excitatory inhibitory balance in each of our subjects. Then each subnetwork is characterized by its own topology. So we computed the intra-FC and sparsity for each of the network, and looking at the results, we saw that intra-FC and sparsity contributed significantly to explain the variance of TVB parameters in our data. And finally, the level of excitation, inhibition, and global coupling in each subnetwork contribute to explain the variation of neuropsychological scores in the corresponding cognitive domain. So we have 3 main take-home messages. This work provides a new framework to perform a simultaneous assessment of multiple resting state network at single subject level. And the correlation between TVB parameters and the intra-FC and sparsity supports the presence of a link between subnetwork topology and neuropsychological properties of brain subnetworks. And finally, considering that TVB parameters have a clinical relevance, this exploration can provide new means to answer pathology-specific questions. So this is a new strategy which paves the way to a novel approach to biomarkers research. So thank you for your attention, and this is my group.